And now, a diagnostic breakthrough for Alzheimer's with Dr. Jonathon Hill.
And we're just getting preliminary data right now, but it's looking really strong to the point where it looks like we can identify not just that there was neuronally derived cell-free DNA, but which neuron type was it? Was it cortical? Was it motor neuron? Was it dopaminergic for Parkinson's disease? And we can tell you exactly the makeup down to the neuron type of the cell-free DNA that was coming from dying cells in your brain. So we're really excited about that. I think we can impact a lot of people. I should mention as well, the DNA signature we found using banked blood was visible 5 years before symptoms appeared. So we have a chance to have a very early diagnostic that can let people know at a time when, the new drugs that are coming out that slow the progression of the disease, et cetera, can be super effective.
Mm-hmm. Welcome to the BioTalk Unzipped podcast, where we unzip the stories behind medical progress by sharing the latest and greatest advances in biopharmaceuticals and medical technologies in a fun, entertaining, and enlightening format. And now your co-hosts, Gregory Austin and Dr. Chad Briscoe. Hello and welcome to BioTalk Unzipped, where we unzip, unlock, and uncover the stories behind medical progress. I'm your host, Gregory Austin from Kansas City. I'm a life science professional in the quality and regulatory space. Along with me from North Carolina is my excellent co-host, Dr. Chad Briscoe. He's a bioanalytical scientific expert, and we publish BioTalk Unzipped twice a month and bring experts from different corners of the pharmaceutical, biotech, and med device worlds here to share their innovations, their insights, career journeys, and personal stories to the show. Today on the show, we have Dr. Jonathon Hill joining us from Utah.
Excellent. Thank you so much for having me on here.
And yeah, absolutely. Dr. Jonathon is a VP of Science and Technology and a co-founder of Wasatch Labs, Biolabs, where he leads research and development for a proprietary nanopore sequencing-based technology with Dr. Timothy Jenkins, who's VP of Research and Discovery and is co-founder. As an associate professor of cell biology and physiology at BYU, he has 18 years of experience with genetic and epigenetic research. His research lab combines bench biology with bioinformatics and genomic approaches to assemble and determine the underlying mechanics of gene regulatory networks. So welcome to the show, Jonathon. How are you today?
I'm doing great. It's a beautiful, bright spring day here. It's warming up finally. The snow's starting to melt. It's looking great.
Excellent. And what we like to do is make a $100 donation from BioTalk Unzipped to the charity of your choice. And your charity is the Rainforest Trust, one of your favorite charities. And I understand that this came from a living opportunity when you lived in Brazil for a couple of years. Tell us about that experience and tell us about Rainforest Trust.
Yeah, so I have a kind of long and ongoing connection to Brazil. I lived there for 2 years during my college years between 2000 and 2002. And I came back with a great appreciation for the people and the wonderful biology and biodiversity that they have there. And as I continued my studies, I kept up my language skills. In fact, at one point, I had a little side gig where I translated scientific articles written in Portuguese from Brazilian researchers into English so that they could get published in the United States. And then last year, I had an opportunity to return to Brazil as a Fulbright scholar. I went down there and worked with the local university there. I taught a bioinformatics class in Portuguese, which was a challenge. But turns out a lot of their scientific words are borrowed from English, so we're okay.
Nice.
And then I got to sequence using nanopore technology the genome of the fire sponge, which is a sponge species that's thought to create several anti-cancer compounds and antimicrobial compounds. So we want to understand those synthesis pathways and what's happening. And so, while I was there, I visited the Amazon and I just fell in love with this wonderful biological resource that we have. It truly is an international treasure. And I think we need to do what we can do to preserve it. As I was telling you guys before the show, one of my bucket list items, one of my dreams would be to set up a nanopore sequencing lab in the Amazon and just sequence that biodiversity and create that data so we could glean it for everything it's worth and learn so much about how our world works.
Just
Nice.
like Sean Connery and his gas chromatograph. And what movie was that?
What movie was that? I'll have to
I
look
don't
that up. That
remember.
was a cool
Yeah.
movie. Yep.
That was a cool movie, other than the gas chromatograph that just made me insane. But
Yes.
yeah, Jonathan, that's phenomenal. And actually, when we were talking before, I was going to save it, but I also lived in Brazil. So
Oh, did you?
yeah, about 10 years
Cool.
before you, I was there when I was in high school as an exchange student
Okay.
in Rio. So yeah, so
Nice.
I've tried hard to keep up a little bit of my language skills, but it's usually when I get talking for a bit, I can get going, or maybe a little bit of a little bit of liquid courage always helps as well,
Yes.
but
Yes.
yeah,
Yeah.
so that's awesome. What city, if you don't mind?
So when I was there the first time, I lived in the southern tip of Brazil in a city called
Hmm.
Porto Alegre.
Sure,
And
yeah,
then this last time, I spent most of my time in Salvador,
in
which is a
Salvador.
beautiful,
Okay,
wonderful
yeah,
city. But I
yeah,
traveled to Rio,
yeah,
so I got to
yeah,
see Rio.
nice, yeah,
And then
yeah.
I got to go to the Amazon, and we spent a week in the Amazon as well. So it was a
That's
wonderful
fantastic.
experience, wonderful
That's
trip.
fantastic. Yeah, my
Yeah.
mom last year
So,
did she did a birding expedition looking at purple martins in Manaus. So
wow. Yeah.
74 years old, and went on a real, it was a pretty hardcore birding expedition too, like knee high plastic boots, traipsing through the jungle and everything
Yes.
else. So yeah,
Yeah, we did some
amazing,
of those hikes.
amazing.
They
Yeah.
don't go light. You're in
No,
there
no,
for real.
no,
Yeah.
no, for
Um,
real, for real.
but
So
it's great.
that's amazing. Yeah.
Então devemos falar português um dia.
Seeing.
That's all the Portuguese I'll
Nice.
speak on here, but
Tá bom. Eu
yeah,
também. Tá bastante.
good, good,
Nice,
good.
nice.
All
So
right.
I was looking into the nanopore technology. It's the real-time analysis of native DNA and RNA, regardless of the length. But in layman's terms, for all the non-geneticists like me, um, how does this technique work, and how does it speed up your research?
Excellent. So nanopore technology is a newer sequencing technology. People call it third gen technology. At some point, I think we're going to have to stop using those numbers because it keeps going too fast. But um, my favorite was when they decided to call things next gen, and it's like next after what, before what. Anyway, it's not a good term. Nanopore sequencing is based on protein nanopores that exist in nature, and these particular nanopores can pass DNA through the pore. So what they found is if you embed this pore into a membrane and then have an electrical current that goes through it, that current is affected by the bases of DNA that are in there. And so each group of bases will create their own kind of electrical signal. And so you have enzymes that are passing the DNA through this pore at a set speed, and you're just reading the electrical current. And as you do so, you can get read sequences. And some really cool things that come of that is that, as you mentioned, it is completely read length agnostic. We get great data off reads as short as 50 bases and as long as 500 kilobases. And it doesn't matter. It will go through and it'll just pass the entire DNA fragment that you give it through the pore. And as it's doing that, it's reading the sequence and it will read until it gets to the end. There is no set length or limitation there. Another advantage that it's given us is methylated bases, bases that have been chemically modified as part of gene regulatory pathways, give a unique signature as well. And so we get native methylation calling from our DNA essentially for free, right? It's just part of the data that we get. We don't have to do any special treatments. We don't have to do any special base calling algorithms, et cetera. It is just there embedded within our data. We use that information a lot in our studies.
So kind of generally speaking, how would the application be different using the nanopore versus Lumina sequencing?
So Illumina sequencing is based on short reads, right? You set a
Hmm.
read length, typically 2 by 150. So you're reading
Hmm.
from both directions, 150 bases each direction, and you're going to get that information. So you're limited to 300 bases per read. Now, I do use Illumina sequencing in my lab research at the
Hmm.
university. It's really great because it gives you a lot of reads. And so one
Okay.
application we do is when we're doing RNA sequencing where we want to quantify gene expression levels, at that point, getting a little tag of 300 bases or so that says, hey, this gene was expressed is all we need. We don't need the entire sequence. But we want lots and lots of reads for the statistical power that they provide.
Okay.
So
Okay.
Illumina is excellent at that.
Yeah.
But if we want to look at context, for example, if we want to look at mutations that are somewhat far apart from each other, but we want to know if they were on the same strand of DNA, or perhaps one came from mom and one came from dad, those kinds of things, we need to have those longer reads. It's also very helpful when you're assembling a new genome, like what we did in Brazil with the fire sponge.
Okay.
And we found it's really helpful when looking at CPG islands that can be several kb long, and we want to see what the methylation state is across that entire island. And so I view them as complementary technologies, right? They each
Right.
have their strengths and their weaknesses, and what they can provide for you. But I've kind of gone all in on nanopore because of the unique questions that it's allowing us to answer that just
Nice.
weren't studyable before, right? We
Yeah.
didn't have the technology to do it.
Yeah. Very cool.
Very good. And with the fire sponge, you mentioned that before, that might have some cancer fighting properties to it. Is that something that your lab or some other lab is currently working on to decipher that?
So in this particular case, we're collaborating with people, right? The lab we worked with down in Brazil, Emilio Lana's lab at Universidade Federal da Bahia, the Federal University in Salvador. They're interested in the reproduction of this sponge species, as well as some of these chemical synthesis pathways, right? And where I came in was saying, hey, look, I have this specialized skill set where I can help get you the DNA and the enzymes that are creating these potential drug targets, et cetera, or drug candidates, et cetera. They're embedded in that DNA, and we can discover what those enzymes are and start trying to map it out if we have that genome assembled. An interesting factor that comes in there is sponges have a lot of bacteria that kind of are symbionts with them. And it's possible that these chemicals are actually synthesized by the bacteria, not the sponge itself.
Interesting.
And so when we assembled that genome, we just took entire tissue, and we're still analyzing the data, but it's likely we have several bacterial genomes in with that sponge genome all together. And using nanopore technology, we can sequence it all and assemble it all without too much issue.
And can you differentiate easily the bacterial DNA from the sponge DNA then? Is that, or is
You
that
can.
not even
There's
necessary?
particular features that will give it
Yeah.
away,
Yeah.
certain replication sequences, things like that. But also, when you have long reads, you get really good assemblies. And so you can get a circular chromosome that
Yeah.
comes out of your
Yeah.
sequencing. You go, oh, wait,
That's
that's
amazing.
not sponge. That's got to be a
Yeah.
bacterial genome at that point. And so
Yep.
you start getting those complete sequences. And it's the fact that you have the complete sequence that gives you the information on where it came from.
Okay. Okay.
Very good. Well, before we get too far deep into some more questions, we would like to kick off our first segment and our first part of New News.
New news.
New News. So recently, there's an article that came out in neurosciencenews.com called Breaking the Genetic Code of Tobacco Addiction. Breaking the Genetic Code of Tobacco Addiction. So this article discusses UCSD's study in which researchers used AI. We knew AI was going to come into this sooner or later, to analyze the genetic underpinnings of tobacco addiction by reviewing extant electronic health records, the EHR records that are out there. A lot of smokers' data to pull it from, so they use AI to collect this quickly. So the study revealed 461 new candidate genes associated with tobacco use disorder, suggesting a strong genetic component to the condition affecting about 1.3 billion people worldwide, potentially. That's the mass of tobacco users. By utilizing the AI in the genome-wide association studies, researchers significantly accelerated the identification process, making it more efficient and cost-effective. And this was all enabled by PsychMerge Network. And they used data from nearly 900,000 individuals, underscoring the scale and potential impact of this genomic research. So they identified several genes associated with nicotine dependence, discovered new genetic factors influencing smoking behavior itself, because very few people will start smoking and then not become addicted to it, right? And so these findings could help personalize approaches to smoking cessation and addiction treatment. So, Jonathon, given your expertise in genetic research and bioinformatics, how do you see the integration of AI technologies like those used in the study impacting the development of maybe novel therapies for addiction, and how might this influence the work being done in your labs?
Yeah, that's a very interesting study that you highlighted there. I had not heard of it before, but what an interesting use of DNA sequencing and AI technology to parse out these patterns. I have so many questions about it, right? Like you said, most people are going to be susceptible to addiction to tobacco
Mhm.
use, right?
Yeah.
So it's almost like the phenotype should have been resistance to addiction, as what they're looking for.
Right.
And I'm so curious about their sample set and how they identified those individuals. Had everybody tried smoking? Right? There's so many interesting questions about it. But I think you hit on the bigger question here, the broader question about the use of AI. And I think AI is both a super powerful and beneficial technology for doing these kinds of studies. And if I have to admit, slightly overused and unfortunately is quickly becoming a crutch, which can create issues and noise, right? The advantage of AI is that it is excellent at finding complex patterns with huge interdependencies, right? Where it's not just do you have A, but do you have A in the complex of B, C, D, and F, right? And all these kinds of things. And that's going to help us understand biology because biology by itself is irreducibly complex. One of my favorite kind of t-shirts, I guess, out there says biology is not rocket science. It's much harder. And the idea is that we can't break things down to single components and understand them. We have to look at them in the context of everything else. And AI is going to be huge for that. But on the flip side, it's also often a black box, right? We don't know why it gets the right answer. It just tends to. And that can be limiting in understanding the biology as well. If we are too reliant on AI and we kind of skip that step of understanding why that signal produces a particular outcome. And I think there's going to be a lot of hesitancy for patients and doctors, et cetera, to pick this up and rely on it because it's a mystery. And so we as scientists in the field need to make sure that we're really parsing out these models and understanding these models, that they're very carefully tested, and that we get better at explaining these models and how they were trained and their function to the people who are going to actually be using them so that they'll trust them.
I think you bring up a really good point there about how does it do it, and it's a black box, and I'm sure the FDA is probably wrestling with a lot of those questions on the data integrity and the data validity of everything that's happening. It sounds great, but is it really real? And we've had a couple conversations on this podcast before about trying to do different things with Chat GPT, for example, and it seems like it's going through all the right steps and gives you the completely wrong answer, and
Right.
trying to sort out why that is. So we do have to be very careful moving forward, don't we?
Yeah, I've had students put in code into ChatGPT and they'll come to me and say, hey, look, I wrote this code to analyze this. And I'll look at the code and say, it's not doing what you think it's doing. And so we have to be careful. We're going to have to very carefully vet these AIs and make sure they're correct.
Yeah, for me, it gives us a start, right? And so when you talk about your students, like, all right, that can help you structure your code, but now you still got to walk through just like you would if you wrote your own and essentially debug it, right?
Yeah,
And figure out what is it doing and is it really doing what you think it needs to do? So
exactly.
what are you using, with your either your university work or with Wasatch? Are you using AI at all to analyze DNA at this point? Is that something built in or something you're implementing? Where do you see that application going for
Well,
you?
we are using AI because all of the base calling models for the nanopore sequencer are based on AI. And
Okay.
so
Okay.
we have to do that. And it's where it's strong, right? You get these electrical
Yeah.
signals coming through. They're changing over time. It's very good at parsing that complex signal, weeding out the noise, identifying what's real, and making those base calling and getting better every day.
Hmm.
Uh, you know, 6, 7 years ago, the error rate for nanopore sequencing was quite high. But now they have really reduced that by creating bigger, stronger, better AI models for this base calling. We also have looked at it for some of the diagnostic work that we're doing. For the listeners out there, Wasatch Biolabs is really interested in using epigenetic signals that we can get through the nanopore sequencing in order to create diagnostics for various diseases. So that's a main focus of ours. And an interesting thing, and I can go into more details how we get there, but we found that if we make sure we have good enough data coming in, we don't need an AI model. The signal is strong enough and it's straightforward enough that we can do some more straightforward regression type models and get our answer without relying on AI. And we really like when we can do that, because now the black box is gone. We can sit down with someone and say, here's the mathematical principle for every single step of this analysis and why we're confident in it. And we can, parse out those signals and look at them individually and really get a strong sense about what our data is telling us.
So you said if you have good enough data going in, what do you mean by that?
So at Wasatch, our big innovation was to create a method that allows us to target specific regions of the genome for sequencing in a way where we preserve the methylation signal. So, there's, hybridization based targeting, et cetera, but they always involve a PCR step in them, which erases the epigenetic signal. You still have the sequence signal, but you've lost the epigenetics. In order to keep the epigenetics, you have to do bisulfite conversion, which has its own list of issues in converting the DNA. And then because you've created AT-rich regions as part of that bisulfite conversion, it affects the PCR efficiency and you create biases. And so the data gets really, really noisy. But we found with our method, we can very accurately, with just a handful of reads, predict methylation signals within a complex set of DNA. Just to give one application, one thing that we're looking at is a fertility treatment where we want to let people know, is there cell-free DNA that's just floating around, not inside any cell, in their ejaculate that contains sperm methylation signatures? And this can be really important for fertility treatments because they might say, hey, we didn't see any there, we couldn't count any, but maybe you do have some, and that changes the procedures that are available. And so we can take that cell-free DNA, sequence it, and down to plus or minus about 1%, tell you what fraction of that cell-free DNA came from a sperm or sperm-like cell. And so, and that was on about 1,000 reads total out of that sample.
Okay. Yeah,
and
that's
so
incredible.
We do that. And the reason why it works is because with our technology, there's no amplification, so there's no noise. There's no bisulfite conversion, so there's no issues there. But also, we are targeting on average, 0.01% of the genome that is actually informative to us. Most of the genome does not help us identify a DNA that came from a sperm cell, right? There's a handful, about 2,000 regions in the entire genome that's going to tell you that. And we can focus in on that. And so we get really clean data, we get accurate data and informative data. And so by taking that information, now we can go into a very basic model and get a very strong signal out of it. So in other words, instead of having AI clean up the noise for us, we're cleaning up the noise at the bench so that we don't have it when we start.
Oh, that's fantastic. Yeah.
That's really good. Could you tell us, Jonathon, about maybe a recent breakthrough or project at Wasatch that you're particularly excited about?
Yeah, we have been working. In fact, one of the projects that was a genesis for this was looking at, could we identify early biomarkers for Alzheimer's? And what we decided to do, instead of looking at protein biomarkers, let's look at DNA biomarkers. Specifically, this idea that you have cell-free DNA floating around in your bloodstream and other body fluids that came from dying cells. It was released when the cell died, and it has the unique methylation signature of that cell type, right? DNA sequences generally don't change cell to cell. They are the same in every cell in your body. But the methylation signatures do change in response to disease, environment, age, cell type, all these different factors. So let's look at that methylation signature and identify cell-free DNA that came from a neuron, a dying neuron. And our early tests were bisulfite and PCR based, but and this paper's out and published, but we already saw a very, very strong signal between patients with Alzheimer's and patients without. In that study, young, healthy individuals had undetectable levels of brain-derived cell-free DNA. Elderly individuals had, 5 to 10%. It was a low percentage of cell-free DNA that was neuronally derived. And someone with Alzheimer's was like 20 to 30%. So that's a big gap.
Hmm.
a very easily identifiable gap that we can identify. And now we've taken that, instead of looking at one locus with PCR and bisulfite, we're now looking at thousands of loci across the genome without those amplification and noise inducing steps. And we're just getting preliminary data right now, but it's looking really strong,
Hmm.
to the point where it looks like we can identify not just that there was neuronally derived cell-free DNA, but which neuron type was it? Was it cortical? Was it motor neuron? Was it dopaminergic for Parkinson's disease?
Hmm.
And we can tell you exactly the makeup down to the neuron type of the cell-free DNA that was coming from dying cells in your brain. So we're really excited about that. I think we can impact a lot of people. I should mention as well, the DNA signature we found using banked blood was visible 5 years before symptoms appeared. So we have a chance to have a very
Wow.
Wow.
early
Yeah.
diagnostic
Yeah.
that can let people know
Yeah.
at a time when, the new drugs that are coming out that slow the progression of the disease, et cetera, can be super effective
Hmm.
and helpful. So
Hmm.
there's a lot of research to
Yeah.
do still. We're not there, right? We're not releasing that yet, but
Hmm.
the preliminary data looks very excited, and we're hopeful that that will continue through and we can get that product out.
And there aren't a lot of great protein biomarkers, that we can use for Alzheimer's. Now, obviously, you wouldn't probably be going down this path, but do you try to correlate the data somewhat to the protein measurements, or is that you're so early on that maybe you're too far upstream if you're asymptomatic?
Yeah, well, and so the data set that we've been working with is really interesting. People have banked blood and then they followed their health records over time,
Okay.
right? And so we can
Hmm.
get blood that was banked, and then 5 years later, using
Okay.
one of the
Right.
protein tests or
Hmm.
clinical diagnostic
Hmm.
tests with the
Hmm.
doctor evaluating them,
Yeah.
we're later diagnosed with the disease.
Okay.
And that's a really
Okay.
valuable tool for us because
Yeah.
we don't have to prospectively collect and wait
Right.
5 years. We've
Right.
already got
Right.
that information.
Hmm.
So we do correlate it because that
Yeah.
is the
Right.
standard when we declare something as, this was an Alzheimer's patient.
Right.
That's how we're deciding that.
Yeah. Yeah. Perfect.
So.
That's fantastic.
I'm curious, when let's say we look into the future and you determine that this is, truly a diagnostic for Alzheimer's, and you can predict ahead of time that this person is either predisposed or they're going to have it, right? How difficult would it be for other labs to, I guess, run that diagnostic, or how expensive do you think it might be? Is that something that will be easy, kind of ubiquitous, that you can get out there to the labs of the world, or what do you think?
So this, I mean, scaling is always an issue, right? And that's
Mm,
something we're
hmm
thinking about because
Hmm.
this is a test that has the potential to need hundreds of thousands, millions of tests per year because it's a screening test that you would do
hmm
regularly, right? Annual physical kind of screen
Sure.
to let you know if you have something coming up. And so we have started from the very beginning to think about those factors. And I think this is important. As an academic researcher, I see far too often where we obsess over just what can we do, and we don't worry about scalability, costs, throughput, those kinds of things. And suddenly we have a test that costs $10,000 to run, and you can run one sample per day, right?
Yeah.
And then we try to say, how come industry is not picking this up? How come it won't go out there and be used? And it's because it's not built for it.
Yep.
So we took a different paradigm. We said, okay, from the very beginning, we're going to make sure everything is automatable on a
Mm-hmm.
liquid handler. We're going to make sure that we can do this in full 96 well plates, right? So that we can maximize our throughput. And with this assay, we have brought down prices to about a tenth of the cost of the current way of doing it, which is just sequencing all the cell-free DNA. By targeting, we can multiplex samples. Everything can be done
Hmm.
on a robot.
Hmm
And so we can just, set up several robots in a row and be processing thousands and thousands of samples. In fact, right now we have a very small lab facility, and yet we estimate that we can run 100 to 200,000 samples a year through that one facility. We're
Nice.
also creating a centralized model. So we don't view this as a point of care kind of technology. The efficiencies are gained in kind of a factory model. And so our model is to have clinical labs collect the blood, and then cell-free DNA is pretty stable in that blood. We've done studies where we leave it out for a week and it's totally fine. And they will send the blood samples into us at a centralized location, and we will run those analyses in a super efficient kind of environment. and then send out those results back to the doctor, and they can talk to their patient.
That's remarkable. So fingers crossed, I'll be rooting for that research to come through, because it's sorely needed. And then we need to get some better treatments, of course, downstream to actually help people. So you mentioned earlier there was some preliminary research that was published. Where was that published again?
So now you got me. I'm forgetting the journal name
Okay.
that it came out in. But I can send you that information. You can send it out to your listeners.
Yeah. Send it to me later, and we'll put it in the show notes, because it would be fun to look at that as hopefully a seminal starting point for this this breakthrough. Yeah, excellent. Thank you for listening to this episode of BioTalk Unzipped. Please subscribe, like, and comment to engage with us, and take a look at our other informative shows.